Okay, we start now. So good afternoon from Bangkok and good morning to Germany. Welcome to the webinar on microbiomes supporting regenerative agriculture. My name is Lily Ervilaitit, Vice President for International Collaboration of National Science and Technology Development Agency or NSTDA. I will be an MC of the event. So as we may all aware that food has become the biggest driver of environmental degradation, food is also the biggest victim of that degradation because while food production can drive soil loss, increase drought and floods and other major weather events, which in turn lower the food production. Food system transformation is therefore necessary to achieve a sustainable food system, and regenerative agriculture can play a significant role in this transformation. And today we hope to learn more from our six distinguished speakers who will share their knowledge under the concept of microbiome supporting regenerative agriculture. But before we start today's agenda, please be informed that this session will be recorded and published on the NSTDA Info YouTube channel, which you will receive the link afterward. Without further delay, I would like to call upon a pre-recorded opening remarks from Professor Prasit Palittapongarnpim, Acting Executive Vice President of NSTDA.
Professor Ulrich Schurr, Director of IBG2 Plant Science Forschungszentrum. Speakers, colleagues, distinguished guests, ladies and gentlemen. On the behalf of the National Science and Technology Development Agency, I am delighted to welcome you to the webinar on the microbiome supporting regenerative agriculture. This webinar to be held in collaboration with IBG2 Plant Science Forschungszentrum, Germany. Nowadays, we have only 10 years in which to deliver the 2030 Sustainable Development Goals. But the impact of our current food system on the nature and climate is limiting our ability to achieve them. Current food system is responsible for resource depletion, diversity loss, and adverse environmental impact. To achieve a sustainable food system transformation is necessary. Additional technology and innovation to protect, manage, and restore nature must be adopted to achieve nature-friendly food production system. Regenerative agriculture is a system of agricultural practice and principles that support biodiversity, enriching soils, improving watersheds, and increasing the capacity of our soil to capture carbon, contributing to the reversal of global warming. In this event, the meeting will establish a better understanding in regenerative agriculture among stakeholders, as well as to highlight an important role of technology, such as microbiome and biocontrol, in the global sustainable food system. Finally, I would like to express my sincere thanks to our German colleagues, IBG2 Forschungszentrum, for the cooperation in this event, and I wish the webinar a success. Thank you very much for your attention.
Thank you very much indeed, Professor Prasit, for your remarks. As stated by Professor Prasit, that this forum is organized in collaboration with IBG2 Plant Science Forschungszentrum Jülich. So I would like to welcome Professor Ulrich Schurr, Director of IBG2 Plant Science Forschungszentrum Jülich, to deliver welcome and introductory remarks. Professor Schurr, please.
Thank you very much, Dr. Lilly, for the very kind introduction, and thanks you also, Professor Prasit, for the opportunity to collaborate with NSTDA on this very important topic of microbiome supporting regenerative agriculture in the context of the UN Food System Summit Science Days. Our current food system is, and we know that in many different dimensions. It's on the one hand in relation to productivity, On the other hand, also with respect to biodiversity, to the environment, but also directly linked to the human health. The call for, and this calls for real sustainable agricultural systems and agricultural system practices which are really sustainable, which are benefiting for the environment, for humans, and for the productivity. This includes the development of new in-balance ecosystems, which are well in regeneration of which are used for production. On the one hand, also to optimize degraded agricultural systems. While crops are often mostly the direct focus of these activities, the health and the sustainability of an agricultural system is strongly determined by the microbes, the microbial communities, and the interaction of the microbes and microbial communities with the crops. Such beneficial microbes have been described in the past, often scientifically, since a very long time, and empirical knowledge also from traditional agroecosystems have been used since mankind started agriculture. So these two different dimensions do we have today. On the one hand, we know that microbes play a big role in sustaining agricultural systems. On the other hand, we also see new opportunities developing from the new technologies which come at hand now for novel technology, which improve the understanding on the one hand for scientific and sound application, and on the other hand also open new routes to scientifically based apply microbes in agricultural systems. And this will be for the benefit of growers, for the benefit of consumers, and for the benefit of the environment. I would very much like to thank the speakers of today's meeting, of today's session, to be with us. It's a very distinguished group of experts in the domain, and I'm very much looking forward to your presentations from very different directions of the microbiome in the agricultural system for regenerative agriculture. I would also like to thank very much you as an audience that you participate today, and hope for active participation in part of this meeting. And I would also like to thank our NSTDA colleagues for the, as usual, common, very professional and great performance in the preparation of the meeting. During the meeting, which I also expect will be, as usual, professional and a very good outcome. And then in the aftermath of that, we will also contribute our conclusions to the UN Food System Summit. Thanks very much for being here.
Thank you very much, Professor Schurr. So today's seminar is packed with very interesting talks, so we should start from our first speaker now. And the first speaker is Dr. Angela Sessitsch, Head of Competence Unit Bioresources, Austrian Institute of Technology. And Dr. Sessitsch has pioneered plant-associated microbiome and is interested in understanding the interaction between plants, microbiome, and the environment. And her group explored the diversity and function of plant microbiota by applying a range of molecular approaches and several other techniques as well. Together with her group, Dr. Sessitsch published more than 150 peer-reviewed publications, and today she will give a talk on food system microbiomes, improving sustainability of food production. So, Professor Dr. Sessitsch, please, the floor is yours. Your mic, please.
Can you hear me?
Yes, we can hear you now.
Okay, so good morning, good afternoon. Thank you very much for the invitation and for the very nice introduction. It's a pleasure for me to say now a few words about the importance of microbiomes in the food systems and how we can use them to improve food production. So let's start with saying a few words about the food system. So the food system is the way how food is produced and how it affects our health, well-being, and the environment. And microbiomes or microorganisms play an important role along the whole food chain. So they play an important role in the whole food system, starting from the primary production until our human gut and human microbiome and human health. Now, microorganisms or microbiomes, so the diversity of and communities of microorganisms as they occur in nature or in any system, they're everywhere in the food system. Of course, well known are those which can cause damage, so pathogens for humans, plants, and animals. But there are many, many, many more microorganisms which are very important, which occur everywhere. So we have highly diverse microbial communities, for instance, in soils, in the plant environment, in all water systems, in food systems, so in the food itself, they play an important role in food production. So in all these different habitats, they play an important role, like for instance, as you will see later on, in the soils or in the plant environment. So it's not only that microbiomes occur everywhere in the food system, they are also well connected and are also important for system sustainability. So for instance, if we look at the link between different microbiomes, we have microbiomes and rich and diverse microbial communities in any soil, so in any environment which is important for the primary production, and the agricultural management and the soil microbiomes will ultimately affect primary production in terms of quality, in terms of yield, in terms of yield security, in terms of also animal primary production. And those microbiomes will then also have an effect on the quality of the food. And also on the safety of the food we produce. But also the taste and quality parameters are affected, and those affect also human gut and human health. And of course, also for the utilization and valorization of waste material, microbiomes are key and very important. Now let's have a closer look at microbiomes and crop production. Quite well known are microbiomes in relation to the human body, especially the gut microbiomes. And it's important that these microorganisms hosted by humans are key for health, for nutrition, well-being, and stress tolerance. Now, it's very similar with the plant microbiomes. So also plants are associated with highly complex and diverse microbial communities in different plant environments. So in the rhizosphere, so that's the soil environment, which is around roots,
Hello.
but also within plants,
Yes.
we have a number of different, highly diverse microbial communities. And also they have a very important functions. They are important for the plant health, for plant nutrition, and for stress tolerance. So in fact, we talk about the plant holobiome. So whatever we see in terms of plant traits, whether it's an agronomic trait or a quality trait, it's not only the action of the plant. It's of course the plant and the plant genome which is responsible for this, but we also have the microbiome, or it's also termed the accessory plant genome, which plays an important role. And the plant invests actually in these interactions. So up to approximately 30% of the allocated carbon is secreted as root exudates. And these root exudates are important nutrients and signaling compounds for associated microorganisms, which again, as I said, play an important role in nutrient cycling and stress tolerance. So what are the key functions of the plant microbiome? So on the one hand, there are a number of functions related to disease control. So many plant-associated microorganisms have the capacity to either antagonize or outcompete invading pathogens. They may also induce a systemic plant response, leading to a better immunity and defense reaction to invading pathogens. They are also very important for any provision of nutrients. So this can be either mobilization of nutrients, like for instance phosphorus, which is very hardly or poorly available, also other micronutrients, but also for instance nitrogen fixation is very important. So that's the only system that provides actually atmospheric nitrogen into the soil and providing it to the plant. So think about rhizobia, so these nodulating microorganisms interacting or establishing a symbiosis with legumes. So this is very important, but we also have some examples of free living nitrogen fixation. Then there is the production of certain hormones which modulate plant physiology and improve plant growth and health. There are a number of other mechanisms related to the improvement of stress resilience, like for instance drought stress or stress to toxic chemicals. Some microorganisms can also degrade toxins, which is also important. And there are a number of other ways how microorganisms can influence plant metabolism. And here on this picture, you see, for instance, pepper plants grown either in a normal soil, and here the soil was amended with a plant beneficial microorganisms. And I think it's evident that this plant, looks better and grows better. And here I show you some other examples with potato and with maize. So here, for instance, these potato plants, so these pictures are all taken at the same, or these plants at the same growth stage. But here the first plant is a control plant that grows in regular soil. And here we have increasing doses of application of a beneficial microorganism. And we also see that the tuber yield and tuber size can be affected. And here you see the results of a drought stress experiment. So here this is the control plant, and here different microorganisms were applied, and you see that these microorganisms have an effect on the stress resilience of the plant. And also here some examples showing the beneficial effect due to the application of beneficial strains. So the potential is huge, and we see really very promising results in the greenhouse. However, we also have to be aware that there are some challenges when it comes to field applications, because there are a number of obstacles. So, and what we need in the field, we really need, on the one hand, we need a successful establishment of any inoculant strain, of any microbiome application. It can be also more complex communities, and a long-term establishment. And in the end, also, of course, the target or the applied strain has to express the beneficial features. And we have to consider that these microbial inoculants are applied into a very complex and very adapted system. So, and the establishment and the competition and the activity of any microorganism will depend on a number of different things, ranging from more abiotic parameters to, of course, also the biotic environment. So there are two avenues how this can be approached. Of course, we have to improve our application technologies in terms of formulations and other technologies to enable an applied strain to really survive in the target environment. But we also have to learn from nature and understand better ecological processes to really predict and identify successful strains and successful applications. Now, the avenues for the future can go in different directions. So on the one hand, we can develop and further develop smart and knowledge-driven microbial inoculants. This can be single strain, this can be microbial communities or simple communities complementing each other in regard to various desirable functions. However, we can also follow different approaches. So for instance, breeding and selection of plant genotypes has so far largely ignored beneficial interactions, or even worse, in many cases, due to the breeding process, many beneficial interactions have been lost. So also there is potential to better breed for plant genotypes which have the capacity to better interact with the beneficial microflora occurring in any soil environment. And the third line is that if we think about upcoming smart and precision farming approaches, so also here we can better integrate microbiome functions. So we can really think about or assess for which application, for instance, or for which crop a certain soil is best suited for. So maybe one soil is better suited for the growth of cereals, whereas another soil is maybe better suited for the growth of vegetables and so forth. We can also direct agricultural management practices based on the knowledge on certain microbiome functions. So there are a number of avenues which can be further pursued. I think it's very evident that microorganisms have huge potential, and that it can be imagined that very successful applications will be coming up in the near future. So with this, I would like to thank you for listening, for your attention, and I look forward then later on to the discussion. Thank you very much.
Thank you very much, Dr. Sessitsch, for your informative presentation. Let me again, maybe I forgot to remind you that the speaker asked to keep your talk under 10 minutes so we could have enough time for our panel discussion later. So our second speaker is Ms. Julia Köebele from Bayer Crop Science, Germany. And Ms. Köebele works in sustainability strategy for Bayer Crop Science, Germany, where she focuses on collaboration with farmers, NGOs, and scientists in order to enhance biodiversity in the agricultural landscape, as well as looking at integrated biodiversity enhancing cropping system. And today she will focus on industrial perspective on regenerative agriculture. Please welcome Ms. Köebele. Please.
Yes, good morning. Good morning, everyone, and thank you very much for the invitation to this very interesting event and for giving me the opportunity to speak here. So as was said, I do work for Bayer Crop Science, and I'm based in the German market. So I do work a lot with German farmers, and I want to give you our perspective on regenerative agriculture today. Starting with the pictures here on the soil, which is the main focus of microbiome farming. Now, we're convinced that we, as was already said in the introduction, we have a common objective, and that is what we call sustainable agricultural production. Now, when we look at the challenges that farmers are facing today, which is climate change, which is biodiversity loss, we need to go into a different production system in agriculture. And what do we understand as sustainable agriculture? We always look at 3 dimensions of economic and ecological, but also social aspects. Now, we think that agriculture as such needs to go into direction of a so-called nature positive farming. And that does mean a system that is resilient and that supports soil health and biodiversity, but that also, on the other hand, produces, as was already said, sufficient food for a growing population. And this is specifically, as I'm coming from Europe, and specifically important in the background we're currently discussing at EU policy level, which is the so-called EU Green Deal, and that is calling for the EU's climate neutrality in 2050. And that is also asking for more inputs coming from the agricultural space for lots of different sectors beyond the food sector, beyond the energy sector. For example, we can think of, for example, jet fuels. We can think of plastics. We can think of textiles, where the inputs in the future should come from the agricultural systems. So that means we need to produce sufficient yields in order to fuel, so to say, all these systems and produce all these inputs. At the end, we would need to achieve a system of an optimum between production and nature. So how do we get there? We think that we need an integrated system, and for that, it's very important that we go for a systemic approach. And for us, it's very important to say that we also need a non-ideologic approach. Thinking in systems does not mean that you exclude one or the other technology, but that you look at what you want to achieve. You would need to go solution and output oriented, and that also means that you would need to go technology agnostic. So whatever toolbox, whatever tools exist, you can choose as a farmer from the tools that are existing in order to produce a nature positive output. Now, for us, it's also very important, obviously, to put the farmer at the center and the farmer's needs at the center of all of this. Now, if we look, I'm going to change the slide. Okay. Now, if we look at farming as a system, you cannot obviously ignore the context that a farmer is working in, and that is the landscape, and that is also the field context that he's working in. So when we look at the landscape and the field context, we talk about natural boundaries that a farming system has, and that is resources. So it's about resource management with a focus on soil health as one of the main, well, the motor of the microbiome. But there are also other functions that you need to look at. For example, as we call it, supporting functions, for example, beneficial, which is also an important driver for sustainable integrated agriculture system. Now, when we look at resources, my company has made a commitment. When we look at our own portfolio in terms of crop protection products, chemical crop protection, we have committed to basically reduce the impact, the environmental impact of our crop protection portfolio until 2030 by 30%. And we're working with independent researchers that are developing models in order to calculate the toxicity and environmental toxicity of the products that are currently on the market, that are currently being used in agriculture systems. And the objective is to reduce mainly those products that have the highest environmental impact. What we're also doing is basically looking already in our R&D processes and in the product development, then we're setting ourselves higher hurdles in terms of environmental resistance, and that we also more intensively look at the impact of those substances on pollinators, obviously, but also on other beneficials, also bringing the soil more back into the perspective. Now, when we look at this part of integrated crop management, I'm convinced that we need to think more in cropping systems. And we have started also cooperation with ETH Zurich and IFPRI, where we're going to look at cropping systems that enhance specifically biodiversity, but at the same time, obviously, also other ecosystem services. That is a very important point, because those systems by themselves will then enhance soil health and biodiversity and also sequester more carbon in the soil. Now, when we look at the practices that are being used in these integrated crop management systems, this is very much going into the direction of conservation tillage. So we're not yet there in Europe, but we're starting and we're getting better. And at the end, it would need to go into the direction of also no-till, and at the end also direct seeding system, which is basically linked. We will also need to go into the direction of permanent ground cover in order to bring back the soil microbiome as a motor back into this and back to work, basically. And all these principles, again, that's where the circle closes, are the principles of regenerative agriculture. Now, what does a farmer need? He needs integrated solutions. And for us, integrated solutions should be an open toolbox that the farmer chooses according to his landscape and field context, according to what he is growing in terms of crops on his fields, and that fits into his crop rotation. Now, we as a company have a whole bunch of so-called integrated solutions. What was mentioned already is precision farming, which is one of the upcoming trends. But I also want to explicitly mention the precision conservation, which I think is also an aspect that is sometimes forgotten. But if we think about crop management systems, we also need to think about what do we conserve? And this goes for in-field and off-field conservation. So if you think about precision conservation in field, you could think about sensors that basically detect what is in the field and distinguish between what should be kept in the field and what is a concurrent to the existing plants and what should be taken out. So if you look at out of the field, obviously you can in a field identify different zones of high and low productivity. And you could also say that a certain part of the field should be spared, for example, for biodiversity measures. And this is a classical conservation farming. Now, we already talked about seeds and that varieties will play a very important role in these systems. There are also new breeding techniques will obviously also play an important role, but also seed density and technologies that are able to identify what kind of soil is in the field, and then basically adapts the seed density according to the soil quality. Diagnostics will also play an important role, so be it pests or be it disease diagnostics, and then obviously the whole range of crop protection products, be it chemical or be it biological innovation, my company is also working a lot in research on biologicals, biological crop protection, that will replace all the chemistry, or those chemistry with a higher environmental impact. That will, and we have already those products in our portfolio, that will improve the soil microbiome, for example, for better nutrient uptake. Phosphorus is a good example, and thereby also reduce the fertilizer input. And we also have products already that support a larger root system, and by that sequester more carbon in the soil. And as a food for thought, maybe also for the discussion later on, I think it's important that we need to start rethinking crop protection, and that crop protection is not a product, but it's a system. Now, just as a last example, for us, collaboration is key in order to achieve sustainable agricultural system and in order to implement and further develop the systemic approach. That is why we have started cooperations, for example, with the Living Soils of the Americas initiative. That's an initiative that is a private and public initiative that was initiated last year by Professor Ratan Lal. We're working also with external universities in order to look at specifically biodiversity enhancing cropping systems. So this can be mixed cropping, this can be certain undersowing or any kind of strip system that we are looking more into, and also see how do our products fit in and what do we need in terms of tools in order to fit these systems and to support these systems. And last but not least, we have currently also started our decarbonization program, where we work with farmers in order to implement the practices that I described, the agricultural agronomic practices, in order to be able to sequester more carbon in the soil. So thank you very much. I'm looking forward to the discussion.
Thank you very much, Julia, for your insightful information. Okay, our next speaker is the founder of Green Innovative Biotechnology Company, Dr. Karsidete Teeranitayatarn. His research and development focus on application of natural substances such as prebiotic and probiotic for livestock and aquaculture, herbal insecticide and pesticide, microbial biological control, and natural plant vaccine or plant Elicitor for crop protection by physical and biosynthesis barriers with the highly unique delivery mechanism. And Dr. Karsidete will deliver a speech on natural plant Elicitor for crop protection. Please welcome Dr. Karsidete.
I would like to share our research last 6 years ago, 60 years, last 16 years ago, focused on research to the food safety, food security, and food sustainability.
Dr. Karsidete, are you speaking? Okay, maybe you have to have your camera off, please, and continue speaking. Thank you.
Okay.
Dr. Karsidete, we cannot.
Okay.
Oh, okay. Okay,
Checking. Okay.
Yes, we can see it now.
Okay.
Thank you.
Okay. Okay. Last 16 years ago, we focused the food safety, food security, and food sustainability, and now we research to the sustainable development goal from the UN. 16 years ago, we support to set up our company research area and office in Nasdaq and Thailand Science Park. We research focus on the plant and animal together. We have the laboratory to solve our customer to produce the new innovative product to solve the problem for the farmer. We focus the global problem to the contaminant of the toxic and chemical to contaminant to the feed, food, and environment. This innovative product is called the natural plant inhibitor. It decreases the risk of plant production and based on the reduced using of the chemical fertilizer, insecticides, and antibiotics. It is the quantity and quality of the cultivated plant. We do the product from the natural product from the waste, from the food, like COS and mineral and amino acid, and it's small molecule, about 60 to 80 nanometer. We use the 5 kilo from Nasdaq and other university in Thailand. And we run the process and run the business based on the BCT economy model. This is the launch from the Thai government to reduce the waste. This product can induction of the resistance of the plant to induction of resistance to defense system with the plant and resist attack from the pests and pathogen such as a fungi, bacteria, and virus, and defense system react from the external attack with the physiological and biochemistry change and generation of the protein and chemical into activation of plant immune system. Elicitor is in the plant, induce the plant resistance. Example carbohydrate, polymer, rapid, glycoprotein, and glycopeptide. This elicitor can induce the salicylic acid, statamic acid, and ethylene. This is the key role of the plant movement. Action can, for the physical barrier, can be forming of the waxy cuticle and increase the cell wall thickness and hardness, reef, and callus deposition and make the spine longer and sharpness. The second is called the biosynthesis position. It can induce plant to release the fibroresin, pathogenic related protein and toxin, digestibility and toxin, or prevent insect and attraction to the natural enemy to kill the worm or insect in the field. And salicylic acid can induce the plant with the systemic acquired resistance to release the terpenol, phenol compound, and so on, and phenylalanine amino acid to induce plants to more stronger. And phosphoric acid is the pathway is called the microgen activated protein kinase and defense against biotic state. This experiment under the water deficit. It can increase cell tolerance and make more survival plant. And ethylene can be induce the cell death, not cell that infected cell, and falling leaf and make a new leaf between 14 days and 25 days. And we have the research with the Synchrotron Research Institute in Thailand. We use the FTIR technique in the plant and animal together. This is our experiment to test the Xanthomonas or lysine. It's a bacterial white pathogen of the rice and infected in the rice. The results show that it can be induced the plant to release and produce the pectin cellulose and hemicellulose in the mesophyll and lignin in the EP. It makes the plant more stronger. And we try our product, this product in the China to test with the citrus screening disease or disease. Most popular of the farmer use antibiotics, example ampicillin, cicillin, in the Guangzhou, China. And the last we research with the ASEAN Herb in Space and NASA, SpaceX, and JAXA, and NASA Thailand. We research with the sweet basil or Thai basil in the ground and on the space. It's launched in the November 1620. And support, company support the student more than 400 schools in Thailand to trial and testing this experiment on the ground compared with the on the space together. This is our collaboration in international and national collaboration. Our passion is solve the global starvation and give the clean food and clean food and prepare for the new era. Thank you very much.
Thank you very much, Dr. Karsidete, for your presentation. Very impressive products. Our next speaker is from a Thai academia, Dr. Suphaporn Chiwatanarak, and she is an associate professor at King Mongkut's University of Technology Thonburi, or KMUTT, and she leads the Microbiome Research Unit of Pilot Plant Development and Training Institute and School of Bioresources and Technology, with a strong multidisciplinary collaboration. The knowledge and tools developed from her team are being translated to agriculture, food, feed, environment, and animal and human health, particularly the application of microbiome technology to economical crops such as sugarcane. And today, she will give a talk on comparative metagenomics reviews microbial signature of sugarcane Phyllosphere in organic management. Please welcome Associate Professor Dr. Suphaporn Chiwatanarak. เชิญค่ะ
สวัสดีค่ะ and hello to everyone. So my talk is going to be the work that we have just published recently, earlier this year. Yeah, and with the motivations that we all agree upon, that right now the farmers in the agriculture sector are using fertilizer and pesticides excessively and result in the ecosystem degradation and climate change. So our group would like to pose a question that, can we restore the ecosystem with microbial functions? So in order to do that, we focus on the Phyllosphere microbiome. As you all know that in plant microbiome, there are roughly 2 major places that the microbes are residing in, on, and around the plant. The more study is the rhizosphere, and the less study is the Phyllosphere. And there is a good review, very, very good review from Gong and Xin, published recently last year from Chinese Academy of Science. They propose that with the harsh environment above ground for something like climate change, insect attack, pathogen, overuse of fertilizer, and soil type, farmers are, could you go back a bit? Yeah. Farmers are destroying the balance of the ecosystem. And in doing so, from the healthy plants, we disturb the imbalance, we disturb the balance of the microbes of the plant and constructing a dysbiosis situation, leading to the loss of productivity and also the disease infection. So it is our focus to study the microbiome of the leaves of the sugarcane to address how excessive use of agrochemicals in terms of fertilizers and pesticides can damage the ecosystem in agriculture. Next, please. So we designed the experiment in such a way that we can compare the microbial profile and composition in the conventional farming and the organic farming of sugarcane. With the sugarcane in the mature stage, around 10 months old, and we use shotgun metagenomic approach to study the microbial profile and the abundance of the microbes in the leaves of the sugarcane. And then we use bioinformatic approach to study the co-occurrence network to see how these microbes are working together in collaboration or in an antagonist way, so that we can look at the microbial profile shift of each farming practice, the shift from conventional practice to the organic practice. Next, please. So the result turned out quite impressive, that in the conventional farming, where the farmers are in the sugarcane plantation in Ratchaburi province in Thailand, we found a lot of depletion of the beneficial microbes and a lot of harmful microbes at the same time with the low microbial diversity. And then when we compare the microbial profile with the organic farming, where the farmers stopped applying agrochemical compounds around 13 years ago, and we found that there are a lot of beneficial microbes, and the microbial diversity in organic farming practice is much more diverse than those in the conventional farming. So we make a question that, next please. What are the microbial profile? If we would like to convert the conventional farming to the organic farming, what will be happening in between the so-called transition farming? So we also work on the microbial profile in the transition farming of another farmers who run the sugarcane plantation and would like to stop applying agrochemicals to their farm. And surprisingly, we saw a shift from the less diverse microbial diversity, and we start to see more diverse microbes in the transition farming. And apart from that, surprised us a lot, that is the enrichment of the toxic compound degraders. We found that these degraders are responsible for the degradation of weed killers and also the insecticide with a variety of types of the agrochemicals and pesticides. So, but when we compare the diversity, you can see that in the conventional farming, we have less diverse, and in the organic farming, when you look at the network on the right-hand side, you can see that there are a lot of microbes working together with a lot of collaboration and connection over there. And in the middle, in the transition farming, you can notice that we start to have more community happening and a little bit more diverse than the conventional farming. But since the owner of the farm just stopped using agrochemical in their plantation just 3 years ago, so we can see the trend that if we stop applying agrochemicals to sugarcane plantation, we can see the beneficial microbes and more diverse microbes appearing once we stop using the agrochemicals. Next, please. So then we have the idea that, so it is possible or not that we can convert the conventional farming to organic farming by using the signature microbes that appears in every farming practices, and we look for the microbes that can have the functions, the major function in a particular farming type. So when we look at the conventional farming, where the farmers apply excessive amount of the agrochemicals, we found a lot of extremophile and also pathogenic species. This reflects that the plant just don't want to recruit the beneficial microbes to the plant and just use the chemical available nutrient that is easily to mobilize from the root to the stem and leaves. Okay, and then when we look at the signature in the transition farming, we found a lot of microbes that can degrade the weed killer in the group of paraquat and also glyphosate, and some other chemical degraders, particularly in the dicolorizing Shivanella and metal-reducing pyrosinase and iron oxidizing leptotrix. And when we compare the signature between conventional transition and organic farming, we see the reduction of the toxic compound degrader start from the conventional farming and transition farming until the organic farming. And the signature microorganisms in the organic farming are those in the plant growth promoting bacteria, for example, nitrogen-fixing bacteria and Lactobacillus plantarum, which have the activities to act as a biocontrol, and Streptomyces also that act as a biocontrol. Next, please. So when we see the shift of the microbial community from conventional to transition to the organic farming, we ask ourselves, in that case, maybe we can restore the health of the agroecosystem back. So we would like to propose that to restore the health of the agroecosystem, we would need to have the core species first. The core species that we found in every type of sugarcane plantation, conventional, transition, and also the organic farming. So with the core species, it means that they are specific to sugarcane. And if we would like to restore the health of the sugarcane plantation that has been applying with the agrochemicals, we need another group of microbes in the group of toxic compound degrader on the left-hand side. At the same time, we would need to knowledge the sugarcane plants with the beneficial microbes. For example, the microbes in the group of Streptomyces, Mycorrhizabium, Bradyrhizobium, Lactobacillus plantarum, Bacillus cellulolyticus, and another group of Streptomyces. Next, please. So in that case, we would like to propose the microbial community to restore the health of an ecosystem sustainably. First, we would need to have the minimal set of beneficial microbes that comprise the NPK provider, biostimulant, for example, and at the same time, with the particular type of farming practice, maybe we would need another set of specialized beneficial microbes. Next, please. So in this case, maybe we would need toxic compound degrader, insect suppression, pathogen suppression, and also the abiotic suppression. For example, the drought-resistant microbes, the drought-resistant enhancer microbe. So in sum, when we combine the two sets of the beneficial microbe together, next please, we can see the collaboration between the two sets of beneficial microbes, comprising the core set of beneficial microbes and the specialized beneficial microbes. And then they have to work together with the link of the common microorganism, so-called hub microorganism in the middle of the picture here. Next, please. So I would like to conclude my work or our work today that if we would like to employ microbiome in supporting regenerative agriculture, first we need to remediate and then regenerate the agroecosystem. And secondly, we cannot use a single strain, but we need to have microbial community to work in collaboration, because this microbial community, they live in a symbiotic way. They can suppress a lot of bad or harmful microbes in the sugarcane plantation. With this, I would like to end my talk with the acknowledgement to the people who have done all the work, the people who go to the field and in the lab and the sequencing, and another important team is the bioinformatics team. And I would like to also thank the research fund from KMTT and sugarcane farmers, the owner of the 3 sugarcane farming that we went to collect the sample from Ratchaburi Prof Thailand. Thank you very much.
Thank you very much, Dr. Supaporn, for your informative presentation. Our next speaker will give an ASEAN perspective on microbial application in agriculture and food production. Professor Dr. Antonius Suwanto from IPB University Bogor, Indonesia. And Dr. Suwanto is interested in molecular genetics and metagenomic study of Indonesian traditional fermented food, biotechnology of plant bacterial interaction, and enzyme bioprospecting from extremophile. Some of his researches have been applied for the development of effective probiotics and prebiotics in agriculture and aquaculture. And today he will give a presentation on microbes on ASEAN agriculture and food production, nutrition and security. Please welcome Professor Dr. Suwanto.
Okay, thank you, Dr. Lily. May I have my slide, please? Because we have a little bit some problem here with the slide. Okay, so I think I just want to give a brief things related with the microbes, and we can see that mostly in Asian we have a similarity, although we have also such kind of diversity too. But in terms of like how we cultivate microbe, that would be something that is most of Southeast Asian country has similarity. Next slide, please. Okay, so our food quality depends actually on the signature of our food microbiomes, and those one might be traced back to the soil actually in the that we are cultivating our microbes. And soils are alive, like fertile soils can contain up to 10 to the 12 or 10 to the 14 bacteria per gram. Next slide. So this soil might reflect on the geography, and this geography might be related, of course, with the climate, everything. But one what is very important too is the microbiome. The microbiome might give a difference in terms of flavor, bioactive compounds that is produced by, for example, a turmeric here. When you plant it in some area, might have bioactive compounds 10 times, 8 times more than if you plant it in the other area. That's not just about climate or the soil fertility, but also might be due to microbiome. Next slide. So the way we do for our cultivation, for example, with the vegetables that is commonly cultivated in soil, might have problem because of decreasing amount of arable land. And this one might you have soilless culture, such as like hydroponic. Next slides. And if we look at, for example, like lettuce here, which is produced by hydroponic or soil ground, this might have a different result. Like what we have been seeing here, they have total aerobic bacteria, enterobacteriaceae, pink pigmented facultative methylotroph, and also lactic acid bacteria. They all less in hydroponic cultivation compared to the soil grown lettuces. So what is the meaning of this one? Okay, so next slide. This might comes to what we have it in our phytomicrobiomes in different style of cultivations. For example, in this case, we have food spoilage, food pathogen, but also we have also beneficial bacteria. So then what are the impacts on our health and regenerative agriculture? This is also we have to address, but because of this one is a complication due to the microbial ecosystem that's involved community, we need a kind of a diagnostic system that is more advanced rather than just culture techniques, for example. Okay, next slide. The other things that I want to mention here is about food that is not just coming from normally we consider as a nutrition. For example, if you drink coffee, then you can see the benefit there. So many benefit to make happy, to make a great health, everything. But coffee is not nutritious like carbohydrate, protein, or fat like this. Coffee is our food, but this is not belong the nutrition that is commonly used. Next slide. So we have actually a lot of these things. For example, in Asian countries, we always think about like carbohydrate, fat, protein, vitamin, minerals. But our food depends entirely on the right side here. The flavor, the color, even the substances like for refreshment, such as in coffee, tea, cacao, food additive, fiber, and what is most important too is microbial cells. This is including probiotics, paraprobiotic, and postbiotics. And in Asian country, we have plenty of examples of this type of things, which is considered non-nutritive, but they are important and essential actually. Next slide. So why is it important? Because like we can see it in this SDG goal to ensure healthy lives and promote well-being for all at all ages. But well-being is the state of feeling healthy, but also happy, not just healthy, but also happy. So the joy of eating, not just only for health, but also must be include the happiness. Okay, next slide. That's why we need to consider that what is happening in the gut, that's also can reflect on the happy brain. And this is already known with the research on the microbiome. That is our microorganism in the gut provide many compounds that can communicate it and affect the brain activity such as dopamine, serotonin, peptides, peptidoglycan, and other things. Next slide, please. Okay, so if we just eat a salad, this one is not just leaf. This is actually we have to understand that actually we have to really know that this type of material contain also millions of bacteria, sometimes even billion. For example, lettuce can have 10 to 100 million bacteria per gram. Next slide. So next slide. So then we can see before this one, you can come back. So many fermented food actually contain more than that. For example, here, soy fermented food from Indonesia, which is called tempe, you can slice a soybean into cake because they are combined with the molds, and in fact, this one also contain bacteria. Every Asian nations love to cultivate microbe as part of their food, and you can find many examples from Thailand, Malaysia to Philippines. They have all fermented food in part of their culture. Okay, next slide. For example, here in tempeh, not just fungus and yeast here. They contain up to 10 billion bacteria per gram of tempeh. Therefore, tempeh can act as a probiotic and also paraprobiotics. Next slide. And for that reason, fermented food are essential for regenerative agriculture. Why? It is important because these bacteria, whether they are dead or alive, they provide important component, not for the nutrition, but for our functional activity. But we require good understanding of the non-nutritive components of food, especially the microbiomes. And this microbiome should be essential for a happy and healthy human, not just happy, not just healthy, but also happy. So you can think about that. Why we need coffee, tea, all of those one are fermented actually. Not to include, of course, like wine and other things like yogurt, such as like fermented vegetable and the other. So we come up here for the last one here, appreciation to a justifiable local wisdom for every culture and every country. Next slide. So in addition to that, also the microorganism, the food microbiome, especially in the fermented food, could provide natural protection against, like for example, in this case is COVID. This is not really studied very well, but many important report for the other diseases provide important clue that they are provide protection. They can train our immunity to become more alert to pathogen. Okay, so next slide. So that's the reasons. Unfortunately, I cannot play the slide, but we just have to understand that the analysis for the microbiome involve a lot of bioinformatics. In addition to the sequencing and the high throughput sequencing, this is not to include like if we study the expression like the transcriptomic or proteomics. Therefore, the whole genome targeted sequencings should be conducted in more deeper understanding for the microbiome analysis. Therefore, as an Asian countries, we should work together synergistically, so then we can unravel the potential of the fermented food, and also how we can include microbiome as part of our diet from farm to fork. Next slide. Okay, I think I just give a brief introduction here on how we can think about our food not just as a nutrient, like a common nutrient, but there are a lot of compound, including the microbial cells, and also how we should treat our product, the agricultural product, the system like the presenter before, that microbiome might affect not just the health of the plant, but also the health of the human, because they come as our food. For example, like I presented to you about the hydroponic compared to the soil-based cultivation. And this is not to include also organic and the conventional one. So this is some important thing to consider. Thank you for your kind attention.
Thank you very much, Dr. Suwanto, for your presentation. And now, our last speaker is Dr. Vanida Kamnert-Phet, Director of Foreign Agriculture Affairs, Ministry of Agriculture and Cooperatives, Thailand. Dr. Vanida experiences include livestock biodiversity, animal genetic resource, and agricultural climate change. A decade of multilateral negotiator in the context of Convention of Biological Diversity or CBD and UNFAO, and a diplomat for Thai agriculture in South Pacific and North America. And today she will share with us Thailand's pathway to sustainable and equitable food system. Please welcome Dr. Vanida. Dr. Vanida, your mic.
Can I share my?
Yes. Did you see the share button?
Yeah, yes. I click it already.
Okay, please wait, maybe. Okay.
Yeah, I try to, yeah, when we first tested, it worked perfectly, but let me try again. But anyway, I will start my presentation maybe without the PowerPoint. Sorry. I see the share button, but when I click it, there's nothing happening.
Have you turned on your file already? You have your file?
Yes, my file is on screen.
Okay. Would you like to send us the file, maybe?
Okay. Oh, sorry. But anyway, I will send it later, so I won't spend much time here. But I will send it in the chat, and then everybody will see it. Thanks so much for having me in such an very influential and very knowledgeable group of people under microbiome. And today, I would like to present the National Pathway for Sustainable and Equitable Food System. I think this one everybody understand what is the UN food system is, because it was announced by the UN Secretariat in 2019 that he will hold a meeting, the Food Summit, in 2021 in September. And the process since then, I think it may be at the global, that they try to identify what is the food system and what the connection between food system and sustainable development. UN is always go for the SDG. So with the many stress and shock, and including the COVID-19 happening, it's really urgent, and people realize that we have to transform. We cannot change now. We have to transform something. We cannot do usual way. So with the food in the focus, food and agriculture, and they say that for the food system can be the driving force that we enable to achieve the sustainable goals in 2030. And the summit is for everybody. And I think we have our Honorable Dr. Margot as in the panel of scientists that look into the detailed side behind all the objectives of 5 action tracks in the food system. But I think what we realize is that food system include everybody. Food system include all activities in the food in the food value chain, from farm to bin. So all the process involved with the food system. And when we look at the whole chain, it includes everybody. So it made up of multiple interconnection. So it's very complex, because when we have to focus, food is for all multi-purposes. Even when with the presentation of everybody here, you look at the microbiome, you can see that it all interconnect, and it can be done by yourself. For Thailand, we in the process of UN food system just early this year in January, because the Ministry of Agriculture and Cooperatives, because we are focal point with the FAO, and all the scientific or paper on the food system is from the Rome-based agency. So that's why it comes to Ministry of Agriculture and Cooperatives to be participate in the UN food system. So we nominate our Deputy Permanent Secretary, Kun Rapipat Jansiwong, as a national convener to contact with the UN, and we get a good cooperation from the UN resident unit here in Thailand. But it is quite a journey when the Ministry of Agriculture go to it, because we always realize that we working in agriculture, we deal with food, and then we do everything in the SDG. But when we try to find and ask that how we transform, how do we change, how will we cope with the COVID-19 pandemic, which is present now, and we are going through it right now. And what happened if there another COVID in the future? So how the sustainable in the food system can really bring us or achieving both the 3 dimensions of sustainability, which is economic, which is social, and which is environment. And I think this one is quite complex to understand, especially when we have all sectoral, we have all different agencies to work on it. As you see that we have 17 SDG goals, it means that all the ministry in Thailand has to responsible one or at least 1 or 2 of the indicators, or maybe more, in order to achieve the whole 17 goals in 2030, which is quite an excessive, I can see. But as the action track, there are 5 action tracks. The first one is about ensuring safe and nutritious food for all. The second one, shifting to sustainable consumption patterns. The third one, boosting nature positive food production. The fourth is advancing equitable livelihood and people involved in food system. And the last one is building resilience to vulnerabilities, shock and stress. And I think the focus that maybe we can in detail in our discussion or collaboration later on regarding microbiome or the topic today is in the action 3, boosting nature positive food production. What does it mean? What can we do? But I think our honorable speaker before me already explained how we can do that. And it seems that they're all science, they're on solution. Now it's have to see how we can make it happen, or how make it work. We're looking to reduce chemical use. We realize the soil degradation. We know the greenhouse gases from agriculture sector, but mostly we're facing smallholder farmers. With our smallholder farmers, the opportunity or the access to capital or resources or the burden of the debt. So it has to look at producing or production community that we have, and we look at the consumer. That's why food system it's all about. I heard that many farmers degrade the environment. I heard that farmers not doing a good things. But when we look at equitable, how they can access, how can they share the resources? I think those one, if we want to transition from conventional to whatever type that we consider sustainable agriculture. I think it has to think interconnected and which policy, which activities can really enhance, can really look into how all of us can do. And I think for the dialogue, for the UN, they separate into the global dialogue, the national dialogue, and independent dialogue. I think this one, maybe Dr. Maragot make it as an independent, which is the science to support all the action in the future. For Ministry of Agriculture, what we do at the national, we do the national dialogue. We talk to civil society through the National Health Commission. And we see that, we recognize that, we realize that we have everything. We have strategy, we have organization, we have knowledge, we have very excellent center for all discipline. But it seems that when we talk about food security, what does it mean? People do different things. We talk about food safety. We invest a lot of budget about research, but consumers still not realize that. How can we do differently? And one thing is that there's so many terms when we talk between a group. What a definition of what we are talking about. I really like Dr. Antonio, Dr. Suwanto, that healthy, it must be happy also. You healthy, and you have to be happy. And I think if the microbiome can make that the solution for nature positive production, not only good for the consumer, but it's good for people all around, it's good for the planet, and it's good for prosperity. And this process, the pre-summit and the global, there are two. The pre-summit will held at the end of this month, 26 to 28 in Rome at the FAO. And our Minister of Agriculture will be participate in that. We will share experience of Thailand. And I think from this, I would like to have the help from all of you in order to share experience and help Thailand to identify what best of our pathway in the future. And I think what we looking at when we go to the summit in September, how we can put food and agriculture in all policy. If you want to put food system in the center of the SDG in 2030, how the all policy, how everybody at all levels can participate. Just like in this microbiome, I can see how the collaboration in the science community between Thailand and Germany, which is different environment, but we can be together, and we can see the microbiome community in the soil and the plant, or even in us in the human gut. And I see this one is how this system can be used and can be transformed. And I think that one, we really open up for any solution that will shape the national pathway, which is Thailand. And one thing that we looking into that through the discussion, we not participate in the global because we do the foreign affair. No, it's not. It's for our next 5-year national plan, the 13th plan of national social and economic development plan, which is in the process. So I think I'm looking how we collaborate, how we can share the experience and knowledges and make the smallholders or make the vulnerabilities, they can be equal, they can equitable livelihood, and they can share value. But one thing I think through the listen to the microbiome, one thing I think the missing from the agriculture or maybe the food producer, microbiome and the value of ecosystem services, which is sustainable agriculture perform. You cannot see with your own eyes. You need tools, you need microscopic to look into what the genome is, what the biome is, what the community it is. You cannot see your own eye, but you recognize, you realize the importance, the benefit. So I think what we can do is that maybe we use that to make to show the consumer and make the farmer knows that if you do a good ecosystem services, you regenerate agriculture, there's a value for that. And consumer or other sector have to value it and give it equitable value. So you can transition from what you say is degradable to sustainable. So I think for the microbiome, you enjoy, the living thing that you cannot see with your own eyes. So let's do that. And I think it's not difficult to transform or to support regenerative agriculture. And our Ministry of Agriculture Cooperatives is open up for all collaboration that the science community can bring to have a good innovation in policy and practices. Thank you very much from me. Sorry for not to share my PowerPoint. Thank you very much.
Thank you very much, Dr. Vanida, for sharing your interesting view to all of us. And I hope that we can continue discussing some of your points during the panel discussion. So now we have completed individual presentations and I would like to spend 1 minute here to ask all audiences to participate in our dialogue report by filling in the small questionnaire appear on the right-hand side of your screen now. Okay. And meanwhile, I would like to introduce Professor Ulrich Schurr, again, Director of IBG2 Plant Science, Forschungszentrum Jülich. And he is now, he will be our moderator of the panel discussion today. So Professor Schurr's scientific expertise is in the field of plant physiology, plant phenotyping, including technology development and quantitative image analysis. He extended this in the last decade to integrated bioeconomy and has established significant activities in biorefinery research, digital bioeconomy, socio-economic research, and strategic concepts towards sustainable bioeconomy. And Professor Schurr has served numerous positions in European organizations, including as a Vice President of the European Plant Science Organization, or EPSO, and as a chair of the European Technology Platform Plants for the Future, which is interfacing between industry, academia, and farmers. He also runs a number of large-scale initiatives in bioeconomy research, like the Bioeconomy Science Center and the Biorevier.com. So it is our pleasure to have him as a moderator of our panel discussion today. So Professor Schurr, the floor is yours.
Yeah, thanks very much, Dr. Lilly, and thanks very much to all the speakers who made great presentations today. I think we have seen a very wide range of different opportunities for the microbiome, ranging from production systems to the food system itself, even to happiness. I think that's a very wide range of things which we can achieve with the microbiome. Because we are already running out of time a bit, I think I would like to pick up as a kind of first round or even final round for the podium discussion, the aspect of collaboration. We've seen that microbiome has so many different aspects with respect for lab activities to field activities, from production to food, from industry to science. So my question would be also looking at the situation which we are facing at the moment with the pandemic, which clearly restricts our interaction on an international level. So what's the view of the different partners, different speakers in the podium now, on the necessity and what kind of specific actions are there needed in order to collaborate in the future? So I would like to start probably with Angela from the beginning, so we go in the same direction as we started, because then Dr. Vanida can actually continue the discussions which you already started with the collaboration, and then probably also go directly into the closing remarks. So Angela, would you like to start about collaboration, the necessity in microbiome research?
Yeah, thank you, Uli. Thank you for the question. It's an interesting question, of course, and collaboration is always important. And if we think about the complexity of microbiomes and the different diversities and microbiome structures and functions in the different environments, I think it's obvious that we have to collaborate to better understand microbiome functions. For instance, we still don't know what is a healthy microbiome, how diverse has a system to be, which kind of functions have to be present, how are they encoded, and so forth. So I think there are different points where collaboration would be highly important. On the one hand, in terms of a better understanding and to really bring together big data, because we will get a lot of benefits if we really bring big data together. And big data is not cannot be made by just or generated by one institution. I think here really a collaboration is needed. But of course, also in terms of to understand the different ecosystems and environmental impacts, so also to link it better to the existing, for instance, plant genetic diversity, which is of course very different in different regions and so forth. and also the technological cooperation.
Thanks very much, Angela, for that insight. Julia, how about you? What do you think about collaboration?
Yes, thank you very much, Uli. As I already mentioned, for us, collaboration is key. And I would like to refer to the presentation of Professor Superpon, who showed this nice example of transition farming between the organic and the conventional. And we are very interested in working with research in order to understand what is necessary in order to reach this phase of transition, enriching the microbiome. And then the next step for us would obviously be how do we make sure that the knowledge of practices, for example, gets also transferred to the farmer, because the big challenge is that sometimes we have a disconnect between science and the results, and everything we're producing great results in science and transferring it then towards the practice that the farmer applies on the field. And here I see big opportunity for collaboration.
Thanks very much. I think that's always the big challenge, to bring scientific results to the farmers and also some farmers' issues and problems to the scientists. So this dialogue is really very important for getting things done in the future. Also between different parts of the globe, between southern parts, between different parts of the globe, because we have different crops, different microbial systems. We can study general ideas, but they have in the end to be actual, they have to be transferred to the real field in the specific zone of the globe. So Dr. Karsidete, how do you think about the need of interaction between different disciplines and the different areas of microbiome?
Okay. Our research is the plant immune system stimulant, plant stimulate immune, and animal stimulate immune. Our research to focus together with the microbiome together. It can induce the pathogenic organism is increase of the fairy microorganism in the soil or in the gut of the animal. I think it's a collaboration with the different of the size. Example, we research together with the
very hard to understand. It's very hard to understand. Sorry.
Okay. This I would like to explain about the plant immune system or the animal immune system to stimulate the animal. The raw material is the Elicitor. It can induce the plant immune system and can to increase of the fairy bacteria on the soil and fairy bacteria in the gut system.
Sorry, it was extremely difficult to understand, at least from my side here. But sorry, it's difficult to understand. Let's probably move on to Professor Superpon. You already showed quite nicely how different activities can move together, and how actually the activities which we have today in the field, how they are changing the microbiome, and how this can be used as diagnostics, as well as how it can be used to be transferred to a new system. So how do you think, what is important with respect to collaboration?
In terms of translating the research findings to the real world, we would need the understanding that we are facing the big problem of the ecosystem that is unbalanced with the end user, I mean that the farmers. And also we would need to be able to have a good collaboration with the fertilizer producer or things like that, as Julia mentioned about. If we would like to transfer our knowledge and to demonstrate that in fact, if we can have a small area and demonstrate to the farmer that actually, if you would like to regenerate your field, you can do it, and we have to work together, something like that. So, and currently at our university, we have a good collaboration with the sugarcane plantation owner that are willing to dedicate a piece of land in their field and let us to do the demonstration. But actually, it would be nice to have a big picture rather than the sugarcane plantation, because the microbiome is plant specific and also geography specific as well. If we can demonstrate that it works in one field for one particular plant, it might not work in other plants and in some other location either, something like that. And another thing that I would like to share for the collaboration is to make a standardized method for everyone. For example, for the DNA extraction from soil, can we have a standard method, and how we can develop the bioinformatics pipeline that we can have the same thing and we can use the same data. So because as Angela mentioned that now microbiome is a big data arena, we shouldn't repeat the same thing because it is very expensive. But if we make a standardized pipeline or methodology, then maybe we can have the data sharing, and perhaps we can set some guidelines that when you generate the data, you should generate in this particular format so that the others can utilize the same data to answer different questions from a different angle. Yeah, that is all for me.
Thanks, Dr. Supapon. This is actually a very important part, the standardization, actually from the project, the EU project, which Angela leads, the microbiome support. One specific work package is about the harmonization and the standardization of activities in the microbiome activities. So that will definitely be a thing which you should have a look at on the microbiome support websites. And definitely, Angela would be very happy to talk more about that, but we are running out of time. So
okay.
Please directly connect with her and the colleagues in that domain. I would like to switch over to Professor Suvanto. So your perspective about integration, you mentioned already a wide range of different things with microbiome, and also about the Asian perspective in agriculture. So what is your perspective on the integration and interaction?
Okay, thank you, Professor Ulrich. Yeah, so I would like to also appreciate you to arrange for such kind of meeting. I think it's very important to have such kind of collaboration, not just only between country, within Asian or other country, but also with the industry, you know, like the research with the industry. But because the research related with the microbiome involve community, the community of the bacteria or the microorganism might need also sophisticated techniques in that case too, sometimes to approach this invisible microorganism, which is cannot be just as a functional like this one. So in the future, I think we should put microbe, the cell, the dead one or the live one, just like what you buy like in yogurt, for example. So then maybe on the leaf, how many microorganisms that might impact our health and happiness. So this type of microorganism which is associated need a collaborative approach to ensure the private sector to put into a label that might benefit us, that we consume certain type of microorganism, and that's why maybe also the reason why we are too clean, for example, to clean then we can can get less immunity. So this is still in the concept or hypothesis, but we may need to prove it by the research. But the research need collaboration. Some research exchange the data or something like that between Asian country and other nations. I think that's it. Very important part that we made. And also, okay, the last one. I would like to convey my best regard to Professor Morakot Tanticharun. That is she always encouraged us to get collaboration, and you see here that because of her we still connect with the group like this. So I really appreciate her. Thank you very much, Ulrich.
Thanks very much, and I can only appreciate the role of Professor Morakot very strongly, because this is also the basis of our collaboration here in between Germany and Thailand. May I give the floor to Dr. Vanida for the final remarks, which hopefully also will pick up a little bit on the aspects of collaboration, which you might also take from the round and the podium discussion which we had now, which you could take to your Ministry of Agriculture to bring to the Food Summit, because I think it's a very important issue, specifically also for the microbiome being so complex, being so important, being so multidimensional. I think that's an issue which definitely requires to be brought to the attention to the broader public and the politicians.
Thank you very much. I really enjoy this collaboration. I think this is the day one for our collaboration. We share results and with a different experience, but when we look at and use the microbiome at the center for discussion, I think this one is the starting point, because when we start with the food system, we talk about food system and then define with our own experience. And what we found, just like I said, that we have it all: strategy, organization, resources. But I think integration mechanism, how science bring to the farmer, I think that's the big question for Ministry of Agriculture too, and how consumer see the same thing as the farmers. If they see the same thing, the healthy one or the happy one is the same definition, I think we don't need to convince, we don't need to say which one is better. I think that one is the same one. So I think the work solution already on the ground. We know what is the good solution. The first collaboration is mainstreaming it. Mainstreaming good solution to the business, to the researcher, to the science, to the policy. I think that's another one. And with the different institution, I think it's fragmented. And we always see individual KPI, which is I working on a KPI. But I think if we look at microbiome and food system and we focus at how you can deliver the nature positive production, I think that achieve one of the food system, and that's the goal, and it is science-based. And everybody here is already science, and we talk to the brain of the sector. So science-based, comparing like Dr. Supaporn showed the community with the different community. I think we need that evidence and science-based and make producer and consumer see the same thing. So science, the connection. Science, the connection, so we can go together and we can transform and go to SDG together. And with a different word, like now in Thailand, SDG, BCG, food system, what does it mean? Maybe we don't need to identify or try to justify we working, but if we do it sustainably and we make it we healthy and we happy, I think that's our goals for collaboration. Just like for the UN Food System Summit say that the summit in September is not the end, it's the beginning. So we still can identify what we can do in the process, and we welcome if any scientific input into the policy at national and the global, and then we can really do. So we're looking for the next step. And my last word is thank you for Dr. Maragot, Dr. Lily Kunsiliwan, Biotech NSTDA, and also the Jülich that allow us to be part of your collaboration, and thanking all the speakers and all the participants. And sorry for not showing my presentation, but it's not impressive as other speakers, so I ensure you that. But thank you very much, and we open up, and I think this is the day one, and this is we start our collaboration. Thank you very much, Dr. Uli.
Thanks very much for these nice, kind words, and I would like to give over to Dr. Lily for the final words.
Thank you, Professor Schurr. So it has been very interesting discussion, and thank you, Dr. Vanida, for your remarks. Very nice. And so before we say goodbye, I would like to thank all of our incredible speakers who give us tremendous insightful information. Thank you, our audience, for joining us today. Sorry that we have no time to give you all the, to address all the questions we have in the chat box, but we will try to send the questions to our speakers, and hopefully they can address and send that back to you later on. I also thank our staff for your well-organized and hard work. And lastly, I would like to thank Professor Maragot Tantijaran, Senior Advisor to NSTDA, for strongly encouraging us to organize this site event, and Professor Joachim von Braun for inspiring us to taking part as a side events to the Science Day of the UN Food Systems Summit. And finally, I would like to ask all participants to register for the UN Food Systems Summit Science Days, which will start on the 8th of July. And the link has shown, here, as in the QR code on this slide, and it can be found also hopefully in the chat box later, maybe not. Okay, only this QR code here. And we do apologize for any technical problems occur, and also that our program might run a little bit late today as well, so sorry about that. But we do hope that you find our site event very useful. And thank you very much again for everyone, and please stay safe, and goodbye.
Thank you very much. Goodbye.
Thank you very much.
Thank you very much.
Lily
ขอบคุณค่ะ
ครับอาจารย์
Vanida
ดรลิลลี่ครับ